I see Thomas Hartung. Yeah. I don't know who is supposed to introduce Thomas Hartung. On the other side, I don't think this man doesn't need much of an introduction. He's very active, very visible in this field, moving things forward. Thomas, I enjoy interacting with you in this mode, as probably many. And it's great that you have the time, that you are available. And I think we don't take away from your time and give you the stage and you start.
Okay, Klaus-Michael, thanks a lot for having me here and introducing me. This time I'm actually speaking from Germany, very unusual for me. I'm at my mom's place and I hope that all of the technology will play. As we are just arrived here for finally a time in Europe after 1.5 years on the other side. Good, so, okay. This is a topic I've not talked too often about. Fighting cancer is obviously inspired by the fact that at the moment there's a beating cancer plan in the EU. And this has an impact on possible animal testing, the need for new technologies. So we are interested to see how a lot of the things we have been doing over the last 10, 15 years are actually coming into play here. Here you can see me as me and most others have been working until most recently. These masks we actually printed. They're for the Center of Alternatives to Animal Testing I'm directing in the US and in Europe. And we wrote this tagline, saving more than animals. And this is a very important message from my side. I'm a physician in the leading school of public health of Johns Hopkins. And if we are trying to promote non-animal methods, this is about improving the situation of patients and consumers. It is not about compromising for animal welfare reasons. This is a breakfast, and I want to use it to illustrate the difficulties we have with identifying chemicals which are producing cancer. Take, for example, here these eggs. Yeah? We are protected, for example, against the dioxin in these eggs. Still, occasionally, we have to destroy sometimes hundreds of millions of eggs because some thresholds for dioxin have been exceeded, which are thought to be not protective enough for avoiding cancer. If you do the exact same animal test with which we derived these thresholds and apply it to the alcohol here in this glass of champagne, you can still drink the sparkling wine. 1 glass in 345 years is the equivalent calculation for the dioxin we are applying in eggs. And this shows you that we are not very consistent in the way we are dealing with carcinogens. We're using a lot of mutagenicity assays, but in the standard assays until most recent revisions, kitchen salt was in some assays a genotoxic agent. So was sugar, regular sugar, when applying the 10 millimolar, which were used to be the upper limit required. On these cucumbers, we have some minute amount of pesticides. They're very highly controlled. We pay about $20 million for assessing the safety, including carcinogenicity assays. At the same time, plants have about 10,000 times higher concentrations of natural pesticides to protect them against insects. And of these, 63 were tested and 35 of them were carcinogenic. So we are bothering a lot about the residual pesticides and their possible carcinogenic effect, but we're not bothering as much about the natural ones. And last but not least, coffee. 32 ingredients of coffee were tested in the cancer bioassay, and 23 of them were positive. So in the US, where we have the Delaney clause, you could not add coffee to foodstuff if it was not a natural substance. So you see there's a lot of things which seem strange. And if you just take the last example, practical epidemiological studies show just the opposite. The more coffee you drink, the less liver cancer, at least, you show, and they're similar for statistics and epidemiological studies for some other cancers. So continue enjoying your coffee, whatever way you prefer. And this shows you that we are really dealing often with shaky information. And it's altogether quite strange that I'm a pharmacologist and a toxicologist. As a pharmacologist, I'm used to studying a single substance with one outcome in thousands of patients. As a toxicologist, I'm used to use relatively small groups of 20 to 50 animals. We assess dozens of endpoints, and we apply in the end lousy statistics. So something does not fit. They are both about a foreign compound, usually something synthetic, and its biological effect on an organism. So if you now look into the cancer bioassay, this is an assay which was standardized in 1970. I was just 7 years old, but almost all of the animal tests in toxicology are older than this. It is an assay which is very costly. It takes you about 18 months in mice and 24 months in rats, which is considered a lifelong treatment, uses 600 animals, and as a consequence, 53% of all substances which have been tested show a positive result. Which does not mean that we have 53% of all chemicals are carcinogenic. This holds even for drugs which are on the market, which have never shown to be carcinogenic. Some 46% of them are positive. Experts assume that it is 5, maximum 20% of industrial chemicals which are actually carcinogenic. And this is producing chemophobia. That's the normal result if people hear, this is carcinogenic, this tests carcinogenic, this is in my shampoo, this is in my food. But if you already ask, how much are they correlating between mice and rats? It is just 57%. And this does not mean that 57% of the cases you have the same tumor. It only says you have a tumor, but it could be kidney in the rat and could be liver in the mouse or something else. And they're dramatically different potency in these species. So showing you the enormous challenges. And it takes about 5 years to get the results of such testing. 2 years of treatment, about 2 years for the histopathology of all of these organs, and then 1 year for reporting and in the beginning for setting up the study. This is not compatible with most product stewardship. You cannot produce, you cannot really make this a standard requirement, even if you could afford this. It's also difficult to make it a standard requirement for things coming to the market because you need about 10 kilograms of substance to run these assays. Tell a chemist to produce 10 kilograms of a chemical which is not yet on the market. And this is showing you that, and we have about 1,000 new chemicals coming to the US market, for example, every year. We will never be able to control this important health hazard by carrying out our standard approach. This is a more recent paper where I summarized some of the approaches together with my co-authors, Raffaella Corvi from the European Commission and Horst Spielmann, now at the University in Berlin. What has taken place in the area of carcinogenicity and reproductive toxicity testing. And actually, there is finally new stuff emerging. At least in the field of pharmaceuticals, the International Conference on Harmonization is at the moment embarking on an exercise to replace the cancer bioassay in rats and mice by a test battery with new things. And I think we need to really adapt these things to food and industrial chemicals. To give you a bit more of background, I want to take an example which has nothing to do with cancer. This was a study which I found really very, very telling. They studied the 10 million residents of Ontario between 18 and 100 years in the year 2000. They took 5 million as the test group and 5 million as the control group then. And they checked for 223 of the most common diagnoses. And then they divided their groups by zodiac symbols, horoscopes. And they actually found in the first 5 million, 24 significant association of a disease with a certain zodiac symbol. And then they could confirm still 2 of them when they used the second 5 million as the validation group. So Leo was prone to gastrointestinal bleedings and Sagittarians to humerus fractures. And this was highly significant. And their conclusion was testing of multiple non-specified hypotheses increases the likelihood of detecting implausible associations. So Or simply said, if you test too much, you get crap results. Because I mean, this was a perfect study. The number of people was perfect. The statistics were appropriate. But the problem is that they asked 223 times 12 questions. And even with these, you get some false positive results. What does this have to do with toxicology? If you take a cancer bioassay, we have only 50 animals per group, but we are assessing 60 different endpoints. And we don't have millions to control for. But it's the same problem of asking too many questions. It's almost unavoidable to get some false positives. There's a lot more to be said about this assay. We're using what's called maximum tolerated doses, which means there's already cell death, necrosis, which promotes inflammation, which is known to promote cancer. The costs are enormous, as we said. Different protocols, 57% concordance only. And there was an interesting study which showed that based on this variability, we have at the moment 53% of chemicals test positive. If we would increase from 50 to 200 animals, we would have 92% of all chemicals positive because we are accumulating even more false positive results. So you can say a chemical which is not positive in the cancer bioassay has simply not been tested long enough or not in enough animals, which is showing you a lot of the problems. So today we have actually moved mostly to genotoxicity assays. So we are moving away from doing a cancer assay, but we sort out substances simply based on a battery of genotoxicity assays. And very important work some 15 years ago by David Kirkland and others showed how little specific these assays are and how we really are producing essentially enormous numbers of false positives, which we then are sorting out and which never make it into any developments, especially not as drugs. So we really need to think about reassessing this properly. What does this battery of assays do? In many instances, then people move into the animal assay and show it was not mutagenic in the in vivo assay, and they can continue working on it. Not a very effective use. So already at my time in ECVAM, we tried to improve the situation, to work towards better mutagenicity assays. We did validate the micronucleus assay and participate in the Japanese COMET validation. We pre-validated the cell transformation assay, which is since then at least partially considered by the OECD. We helped reducing the false positive rates by improving the cell lines and the maximum concentrations used. And we also showed that we can omit the positive control in vivo studies. So this was a very comprehensive package which was moved forward at the time, and I think had impact on how mutagenicity assays are being done. And our list was long. This is what we recommended, what we were doing. I don't have the time to go into this, but this is really about improving an assay, which is the prototypic assay where the key event for cancer, in this case mutagenicity, can be done with better assays or combinations of better assays. We have then discussed in 2012, and this was a very comprehensive work with 35 experts in a long process. We addressed systemic toxicities, among them carcinogenicity. And the recommendations of this group was in the end for carcinogenicity to abolish the current test because there's enough evidence that the animal test is not serving us, to optimize the genotox battery, as I just explained. It recommended to move on in evaluating the cell transformation assay, which is also possibly, it might be possible to humanize this, so to move away from animal cells to human cells as the basis for cell transformation. And then to develop integrated testing strategies, which are including the non-genotoxic modes of action. So a combination of in vitro tests, so exactly what is now taking place under the auspices of the International Conference on Harmonization in the Drug Field. And also to assess whether the mechanistic approaches of toxicology for the 21st century could form a basis for identifying pathways of toxicity and improving the battery of integrated tests. So here you see the hallmarks of cancer development. It might be a little small to read. I want to be quick here, but we discussed, can we define thresholds of toxicological concern, or even better, internal thresholds of toxicological concern, so plasma levels of substances which are raising a concern, that we can simply say substances which do not reach these levels, they cannot really be a problem. We talked about the battery to be optimized both for the genotoxic mechanisms and, very importantly, for the non-genotoxic mechanisms. And if you read this here, there is aspects like inflammation, endocrine disruption, reactive oxygen species. So a lot of things for which we have really very nice in vitro assays available, which simply have not been used in the context of creating a battery of assays, which together could give us a comprehensive view on is a substance possibly carcinogenic or not. We have not really ideas yet how we can use metabolism and inactivation as a consideration, but since this would only lower a possible hazard, I think this is of lesser concern. And then there's the cell transformation assay. We were blown away in the end that cell transformation assays almost by 90% predict, after a careful review was done by the OECD, almost by 90% predict the outcome of the cancer bioassay in rats. It is an astonishingly efficient assay. Unfortunately, nobody knows why it is that good. And this has led to limited use. And I also cannot tell you that the cancer bioassay in rats and mice is not a good assay, and then say something which is 90% predicting it is a good alternative really. And all of this should be combined in integrated testing strategies, IATAs, or defined approaches, as the OECD is calling them now. So a way of not using a single assay, one suits all, but something which is being combined. There's also computer-based methodologies. This was an article in 2009. I wrote about the topic, and interestingly, one new kid on the block was missing. We did not even discuss AI in this review paper here with Sebastian Hoffmann. And the reason for this is that AI really came into broad use only after 2012, when deep learning came up, and the computer power was actually getting good enough to produce really good AI models. Before this, you can see here, this is expert judgment and various computational tools in a review article from 2003. And you cannot see there's nothing which is really extremely sensitive or extremely specific or really remarkably different to others. So there's some limited usefulness of these earlier types of computational tools, not very different from just the feeling of an expert. Interestingly, AI was in toxicology first applied in this paper from 1984. At least this is the earliest paper I could find. And Herbert Rosenkranz was really in this paper already describing a method to identify carcinogens using machine learning approaches, 1985. In his paper only 5 years later, he already gave pretty good numbers where substances which showed carcinogenic effect in the National Toxicology Program cancer bioassay were predicted with almost 100% accuracy. So it is possible really to develop systems as already these early precursors for artificial intelligence have shown. Just as a footnote, his work extending this also to developmental and reproductive toxicity is one of the early research grants CAT gave out. So we were supporting this type of work. And there's now really a lot of new tools coming up because we are suddenly dealing with big data in toxicology. These are not only the legacy data of the past, which make some 3,500 cancer bioassays available. We have internet data, the scientific literature. Take PubMed, 2.5 million articles per year, and 80% of them describe some substance effects. So 2 million articles are just put into PubMed every year with effects of chemical substances on living systems, sensor technologies, robotized testing like ToxCast and Tox21, the various omics technologies, high content imaging, these are all producing gigabytes, sometimes terabyte of data from our experiments. And AI can help us to interpret these. So the message so far was, we are not 70 kilogram rats, but we should also be clear, we can do better because we're not 70 kilogram cell cultures either. But there's an opportunity to move really towards highly sophisticated microphysiological systems, organs on chip type of technologies, which are helping us to study these things now in non-animal systems. I have to refer you for technical details to workshop reports like these, which we produced with some 35 experts in each case. The last one was published in 2020, which describe how cell culture over the last decade has really changed from these pen-fried eggs in a pen, sunny side up, to reproducing organ architecture, organ function, and also adding very often perfusion and some hemostasis to these systems. And expectations are high. To give you only one testimony here, this is Francis Collins, the head of NIH. And he, when testifying to the Senate about his budget, already in 2016 said that he predicts showing one of these lung-on chip models, actually 10 years from now, he predicted safety testing for new drugs and environmental exposures will be largely done with these type of organ-on-chip things. So it's just 6 years left or 5 years left, but you see the expectations are really high here. To give you an example from our own field, we are working with brain organoids. This is one of these so-called mini brains. And in the cancer field, for example, we are working towards personalized medicine. So we took glioblastoma, brain cancer, and grafted our mini brains, as you can see here, with the dense materials, which is expanding in the mini brains, showing you glioblastoma in the system. So we published this in 2019 as one of the early examples of a organoid grafted with a tumor. And this allowed us to show then that this can be used for drug development and possibly in the future also for optimizing as personalized medicine the choice of the chemotherapeutic for a given patient. Now let's think about how can we actually protect humans. And this is a very interesting study which was published in Science by Bart Vogelstein, one of our very famous physicians here in Hopkins. And what he showed was really interesting. They did a very intense study and showed that hardly any cancer is hereditary. Most of it is just by mistakes in replication, so chance, and a very small portion is actually environmental. So these two are bad luck. You cannot really protect against these cancers because they are the machinery, you can only treat the cancer. But the 40% they identified which have to do with environmental conditions, with the way we are living, these are actually interesting because here we can modify exposures and these 40% we can actually beat relatively easily in comparison. So this is about prevention and harm reduction. Prevention would mean we simply don't do it, and harm reduction would make the products safer. So for example, a safer cigarette, as you can see here to the right, because this is still one of the key issues for cancer. If you see here a relative evaluation of cancer risk factors, you see that the chemicals which we have been discussing so far are at the lower end as contributors to production of cancer, according to what we know today, but the cigarette are still the #1. And here you can see quite impressively how the consumption of cigarettes in blue is followed by the cases of lung cancer with about 15 to 20 years of delay, which is simply the time for developing the cancer. And you can also see how the decrease in smoking already leads to a decline in lung cancer. So there's a very clear connection here with this still being the most common cancer in men. So the big thing is actually to tackle smoking. 1 billion early deaths were predicted by the WHO for this century, or other way, if you start smoking with 18 and you smoke 18 cigarettes per day, you live on average 10 years less. This is very clear data. Here's where we can tackle. And we have really some possible game changers here. The electronic cigarettes, or more general, the non-combustion tobacco products, are taking away at least the components which are producing most of these cancer effects. Experts agree largely that they have about 3 to 5% of the risks of cigarettes. So they are really a considerable harm reduction. And I've been talking and publishing on this topic quite a bit. This is here from the Scientific American at AAAS and others, because from a public health perspective, they are interesting, but they're also a challenge to toxicology, as you will see in a second. Here you see the increasing use over years. So this is a product which is taken up in Europe, but also elsewhere. There's more and more use of these. But there's a perceived problem, which is kids are using it. Especially here in the US, we have tremendous discussions about the risk of an epidemic of children, shown in red, taking up the smoking taking up vaping, while we see that at the same time, there's a continuing decrease in actual smoking in these children, which is the desired effect. And even though it's only 3 to 5% of the risk potential, it is still worrying if these kids start smoking and vaping electrically. This is an article where I then summarized what is the role of possible test methods in this field. It's all available in Altext, our open access online journal. And the interesting finding was really that already in 2014, there were 7,700 flavors on the market. And in the study period, they observed about 200 new flavors coming to the market every month.
Thomas,
yep.
You have 2 more minutes?
I'm almost done, yeah. The problem is that essentially none of them have been tested. 75% of e-cigarettes, for example, include diacetyl, which you know as the buttery flavor of popcorn. This produces popcorn lungs when inhaled, not at the levels we find in e-cigarettes, but this means that substances which are perfectly safe when ingested are not necessarily when inhaled. So you can see we would have to control this. It's a very costly animal test if you really wanted to address this, but this could produce enormous testing of flavors. And as predicted, these flavors then showed, or these additives showed, with the example of the cannabinoid oils which were added, which led to a small pandemic and a few deaths already in the US. This is now calling for the use of other type of approaches. And I think I'm bringing Cole to Newcastle here when saying that lung models are a primary thing to be done. I believe that we should consider using them for mutagenicity testing, for implementing the non-genotoxic mechanisms to control these, and to consider a humanized cell transformation assay in these models. And this is leading me to my last 2 slides. This is a study where we have been using these type of models to address not vaping, but in this case, water pipe smoke. One water pipe smoke session can be the equivalent of 180 cigarettes smoked. So it's really a very intense way of exposing. And this is a lung of chip model, which our colleagues at the Harvard Wyss Institute of Don Ingber are using just to show you the potential of really moving forward. And I believe that we cannot wait for clinical type of trials to help e-cigarettes to be safety assessed and find their right place in the strategies to prevent cancer. Thank you very much.
Thank you. It went quite well from home. The internet was stable. Nice presentation. And I guess there are questions, Samuel, that works best if you do that part.
Yes, thank you very much. Again, it was like usual, a great talk. So, so it seems like your presentation was pretty much clear, since there was no open question at this stage. However, I would have one to start with. Is that do you think personally that the short shelf life of the in vitro model is a limitations for carcinogenicity, especially considering the self-repair defense mechanisms of human cells. What is your opinion about that?
I mean, first of all, lung models are the perfect example that you can have actually long-term shelf lives and long-term exposures. I also think that most of the mechanism, this is part of why these models are appealing. They are short-term mechanisms, as you know, as long as you have really the key mechanisms reflected, which are promoting cancer, which are mutagenic. I think we can work with short-term assays, and this would actually make it so much more testable if it is not, again, a battery which is accumulating false positive assays. And to some extent, I hope that a better integration of these findings, of these measurements by artificial intelligence, evidence integration, will help us to actually come to a very efficient battery in predicting cancer with short-term assays.
Thank you.
I see a question by Otmar in the gallery.
Right. Hey, Thomas, good to see you. Thomas, your cell transformation assays really seem to be a potent tool. Could you elaborate a little bit what you mean by those assays?
Okay. There's essentially two variants. One is based on 3T3 cells. The other is a primary cell, which is irradiated. They both show pretty good performance. These hamster kidney cells a little bit better. There's a Japanese variant of the 3T3 assay. They have been all pre-validated, evaluated with background review documents. And what you're observing is that in the presence of carcinogenic substances, the cells stop growing, stop their growth inhibition and go beyond the monolayer culture. They're building small hills of cells because contact inhibition is lost, and which you can then identify as transformed cells. Astonishingly good correlation. Nobody really can explain it, especially as some of these are not really very metabolically competent as they would need to be for our understanding. But more than 400 substances have been found in good studies, which gave, as I said, almost 90% predictive qualities. It's a very costly in vitro assay. So in vitro is not cheap necessarily. It's more than $10,000 for one chemical to run a cell transformation assay because you need so many cell cultures and long treatment times to observe this.
Oh, that's a severe limitation. Yeah. Oh.
Yeah. But nobody has really tried to find now, let's say, genetic endpoints, high throughput type of measurements, optimization. I think there's a lot of potential in these assays to do them better and to humanize them. Yeah.
Okay.
Maybe one last question from Laura Gribaldo. Laura, if you want to.
Yes, since it's a pleasure to see my former boss here, right, Thomas?
Yes, so, Friday I will come to Italy finally. So
wow, so we will meet again. Okay. Now, Thomas, since you are here, I profit to make you a question about qualification validation of models, because as you know, we did now this collection of models for biomedicine, and we are frequently challenged by the scientists, the audience, about which method we recommend, which characteristics should have a method to be a model to be applied fruitfully. And so the question frequently is, should we validate this biomedical model, or should we qualify, or which criteria or which performance indicator we should use to standardize at the best this model? Have you any suggestion for this?
I mean, first, I think there's no alternative to validation, because this ensures that decisions are taken on solid ground, and there's nothing worse if you take decisions on bad assays. The problem is how we do it. In the beginning, we have been spending roughly $1 million per assay and 10 years to get a validation done, which is not applicable to methods which also are still developing, which are highly dynamic. So we need to find some shortcuts here. There's no shortcut in assessing reproducibility. This is something which needs to be done because that's the first thing. But you really need to think how we can more quickly identify whether something is relevant. And here, I think we can really move to mechanistic relevance, showing that something is reflecting the scientific knowledge of today, which is more important than showing that it is reflecting a single animal test or so. And I think that we could automate some things here, thinking of systematic review type of analysis, showing the relevance of a given assay. So this is a very, very condensed answer to something we both have been thinking about the last 20 years.
Okay. Okay. Thank you, Thomas.
Mhm.
Thank you.
Okay, it was a pleasure. I hope that someone will have an in-person meeting I can join.
Sure.
Absolutely. Is there still time for a question or a comment? Samuel?
No, there is I think we have to close now, and I warmly thank all of you. Thanks Thomas for the great talks, and also thanks Klaus, Michael, and Laura also for chairing this session. So we will have a break now. I strongly recommend you to visit, the poster platform through the search session on our website, and also to attend the QMO space. This is let's say a specific networking program that we will soon receive a link and potentially in the discussion box. We will meet again at 1 p.m. this afternoon to start with the afternoon session. Thank you very much, and see you soon at 1.